This meeting is being recorded.
In this last section of Chapter 15, we're going to talk about a specific type of carbon-13 NMR spectroscopy, this DEPT or DEPT spectroscopy, which stands for this distortionless enhancement by polarization transfer, but you can just know it as DEPT spectroscopy. And we know that with carbon-13 spectra, we would have these decoupled spectra produced, and this gives rise to singlets that really do not provide any information about the number of hydrogens attached to each carbon. So we know the shift, and we have each distinct carbon producing a signal, but without another type of spectroscopy, specifically this depth enhancement by polarization transfer technique here, we can elucidate using different angles. So we can see with depth 90, we can have a signal appear for our CH here. So carbons with that single proton. Then with depth 135, we can produce positive signals specifically for, again, CH carbons, and then carbons that are part of methyl group, CH3s. And then we also have with this depth 135 a negative signal produced with each of these CH2 groups. So again, each distinct type of carbon now, using this depth technique, will produce different signals, either only CH signals with a depth 90, or CH signals and CH3 positive with the depth 135, and then negative. So we can summarize this here in this table. So we see our normal broadband decoupled carbon-13 NMR spectroscopy would produce all singlet, all positive signals, if you will. So all of these signals up top here, whether it's a CH3, CH2, CH, or a quaternary carbon, would produce the same type of signal. But then we can see with the depth 90 here, we get that positive signal only for CH carbons, but then we overlap that with our depth 135. So all three of these types of spectroscopy would produce the same signal for this CH type of carbon. Then we see with our broadband decoupled, that's the only one that produces a quaternary carbon signal. So we can see either all signals produced, or only the broadband decoupled with the quaternary carbon. So those are ways to look at all three of these spectra in combination and start to build our molecule from all these details of these different types of spectra that overlap. And then we see specifically with our CH3 carbons that we would have a positive 135 signal here with this depth 135, whereas with any CH2 carbons, that would produce a negative signal. So again, in combination, we can build our structure of our compound. And then on this last slide here, we'll briefly talk about one specific application of magnetic resonance imaging here, which is, as they describe, essentially a proton NMR spectrometer. But instead of a sample in an NMR tube, the body is analyzed. So that's our sample. And different tissues have different concentrations of protons in different environments. And this magnetic resonance imaging that's produced is a 3D image of different tissues. And we utilize, again, a magnetic field to orient all these protons and then pass a radio wave through them. But note here that these are mild and no side effects are generally experienced. So again, this is just a real-world application of proton NMR.